Welcome everyone to our Cellular Conversations, a new webinar series. And today my name is Marta Gabrielska. I'm from Millennium Science. We are a distributor of 10x Genomics. And today I have with me also Jerry Ma from 10x.
Hello, everybody. I'm Jerry. I'm a senior field application scientist at 10x Genomics. I'm here to do all your technical support. Back to you, Marta.
No worries. So idea behind this series, it's very much based on the similar conversations that we had about spatial technology from 10x Genomics, but we all believe that single cells absolutely deserve a spot, and we all want to talk about cells. So the idea is that this webinar is shorter. It's more focused on the users of the technology, their perspective, their experiences, how it works in their hands, what advice they can give you. And we seriously encourage lots of conversations and questions. Regarding questions, feel free to put them in the chat during the presentation. We will read all of the questions. You can also raise your hand and you can ask a question yourself. And with that, I would like to introduce our speaker, the fabulous Dr. Jennifer Boer from RMIT University. Jennifer used a lot of single-cell sequencing, and today she will present about her experience in relation to clinical trials, some nanoparticles, and what happened with the good experiences, not that great experiences, and what are her plans. Jennifer, the audience is yours.
Thank you so much, Marta, and thank you for inviting me to this talk. I shall start with sharing the screen. Awesome. All right. Hello, everybody. My name is Jennifer. I work as a senior researcher in the Cancer Aging and Vaccine Laboratory at RMIT University in Professor Magdalena Plebanski's lab. So first of all, thank you so much, Martha, and everybody for inviting me. I was super happy that you'd started this new series of cellular conversations. Because most of all, and obviously to be invited to, speak a little bit about my own experience with single cell data. Now, I think that everybody in this forum can agree with me that single cell data is exciting. And we can see that usually through all the, the lovely stories and exciting findings that we will find at conferences, at talks, on posters, and, high impact papers, et cetera, et cetera. The only thing is that what we usually get to see is just the finished story. We never actually get to understand or to get some insight in what happens in the background of all of these people that embark in a single cell journey. And so this is kind of what the premise, what the purpose of my presentation is today. I won't show you a finished story, but I will show you a little bit how, try to give you a different flavor of how different workflows can provide different outcomes on, what your single cell journey ends up being really. And so today we're going to space from clinical trial to preclinical nanoparticles. And we're going to go on a single cell adventure in which we will look into a little bit of the good, the bad, and obviously I'll give you a bit of exciting as well. Now, in the Cancer Aging and Vaccine Laboratory, we deal with many different types of data and also big data. And, as we all know, single cell can be really useful in answering questions that, that are resulting from lab in vitro results to better understand clinical trial outcomes, as a result of some exciting bulk RNA sequencing outcomes. We've used it in conjunction with shotgun metagenomics, so microbiome profiling, and also in stuff like methylation profiling to, for example, identify transcripts alterations in key genes. Now, for the purpose of this presentation, I'll shortly introduce you to two different projects that we're working on. We won't delve too much on the details of the projects, but I'll just for the purpose of helping you understand what the premise of those projects were and how we embarked in our single cell adventure in these areas. So, One is a clinical trial, and this is the vaccine immunomodulation throughout the aging lifespan trial. We call it VITAL clinical trial, and this has different arms in which different age groups and genders get vaccinated differently. And the premise of this research project is that as we grow old, so does our immune system. And we undergo a process called inflammaging. And so when the inflammaging process starts happening, B cells and T cells start acting differently. And so does, for example, our reactions to specific types of vaccinations or combinations thereof. The other project is a preclinical evaluation of nanovaccines. This also had a very simple setup. We have different treatment groups and one PBS group. And the premise of this project is that, generally we deal a lot with whole organism vaccines, and these are very effective. But the majority of our vaccines tend to be subunit vaccines, and these are usually protein and peptide vaccines, and they're very safe, but not necessarily very immunogenic. And so they usually require some sort of an adjuvant delivery system. So in our lab, we very much believe that the future of vaccines also lies in developing the use of nanotechnology. And so for both of these projects, we embarked on a bulk RNA sequencing adventure. And so for the clinical trial, we asked ourselves, well, does patient transcriptomic profile give us any insights into the immune system and any responses to vaccination? And for the preclinical study, we wanted to know whether there were specific mRNA signatures that we could leverage to better understand these nanovaccines. And now we got to a point where it was all very exciting. And this is usually where you get the very polished story, right? Well, we had the vaccines and there were different gene signatures and all sorts of stuff was happening. We could see that the signatures were clearly really different between males and females. And the same was happening for the nanovaccines. Specific genes were contributing to very important pathways within the biological context that we were studying. And so we wanted to know for both of them, so, all right, well, what is happening specifically, between females and males and the older and the younger cohort? And in the nanovaccines, we really wanted to know, like, what's happening in these specific genes that are informing these pathways. And for both projects, we were really asking ourselves, well, in what cells is this happening? And so usually when you get to the point that you're asking yourself, well, are summary statistics hiding something interesting, then it may be time for a single cell experiment. And so what we did, we started single cell RNA sequencing, and in the clinical trial, we asked ourselves, all right, well, what cells are changing the mRNA signature profile when our aging cohort is responding to vaccines? And in the preclinical trial, we really wanted to know what specific type of immune cells were responding so well that was mounting the exact right immune response that we needed in this context. And so here, I'll take you a step back, not to the results, but to what our workflows were, because we had very different challenges within the workflows themselves in setting this up. Now, for the human clinical trial, we had frozen PBMCs, and so the blood cells were, in the vapor phase. And so the only thing that we really need to do is take them out of the vapor phase and then start cell prep, cell count, and our gem formation was all in the same area. For the mouse project, however, this was very different because the mice needed to be immunized at certain time points. Then, we needed to harvest the lymph nodes all together. And so you had to do a lot of prep to organize that and to also within the consideration of batch effects, et cetera, et cetera. And then, so we needed a team that would be harvesting the lymph nodes. And then we needed the team that needed to start the cell prep in a completely different location. So with the mouse study, we're dealing with cells that are fresh, but we don't want to be, we didn't want them to be sitting around for too long. And so we had to really organize ourselves around the logistics. Whereas with the frozen PBMCs, the logistics weren't really the problem, but it was the frozen cells that, could have created a bit of a problem. Now, for any of you who have already started or embarked in a single cell adventure, one of the major conversations that you would have had with the 10x expert team is that the quality of your sample the cells need to be of very high quality. And so what does that look like, right? We have low quality, usually looks like a lot of debris, dead cells, and aggregates. We have a medium quality that kind of looks like there's a little bit of debris, dead cells, and some aggregates, and usually a high quality is what you want to aim for. And this is defined as ideally over 90% viability, but we can reach, 70% is said to be acceptable. And really, the minimal cell debris and cellular aggregate is what is very important in this context. Now, usually, if you reach a point in which your sample is of extremely low quality, you have a lot of debris and aggregates, et cetera, 10x Genomics highly recommends the abandoned ship. And now, well, this is very understandable. If you, however, are dealing with, for example, samples that are coming from various hospital samples that are historical samples, which was our problem in this case, then you tend to not really want to abandon ships. Specifically, some of the PBMCs that we had were longitudinal studies that had started from the 90s. So you're dealing with historical samples that are rare types of diseases. They're historical, they're longitudinal, and they're pre-COVID. So at this stage for us, we had very low quality sample, and this was probably mostly due to the freezing sequences that had happened in the past. So really nothing that you can do about it, but also now you have a problem that you cannot abandon ship, so you have to continue. But the problem is the Chromium controller is a microfluidic system. And so if you load this with an amazing amount, an incredible amount of debris, then you're either going to ruin the machine or your gem formation is going to fail. And so at this point, not only have lost your sample, but you've also either lost the machine or lost your precious single cell reagents. And so we had to very quickly switch our mindset. So abandoning ship wasn't an option, but we needed to pull through, and we needed to pull through in a way that would put us in the best possible position to continue. And so what we tried to do is just think about, how do I minimize the chances of ruining or losing the reagents? And so we just had to proceed with trying to get the most amount of debris and dead cells out of it, and continued running it. Now, for usually when we see all sorts of results out there or description of how to continue with bioinformatics analysis, et cetera, it's based on the premise that you have a high quality sample. But what does it actually look like when you manage to still pull your sample through, but obviously at a very low quality? And this is, the first step to kind of see that is your barcode rank plots. And so your barcode rank plots are really nothing else than an indication on a graph of how the cell calling algorithm was able to distinguish between your intact cells or your background barcodes. And so if we look at our first figure, we can see that what you usually want a very steep cliff, and that ends up in a plateauing knee. And the steeper the cliff and the better the plateauing knee following that, the better the cell calling algorithm will be able to distinguish the intact cells. And you'll also see a very nice separation as well on your t-SNE plot. Now, if you have, kind of a medium type of cell, you can already see that this very quickly results in a cliff that is much less steep, and the knee that also tends to plateau less optimally compared to the high quality cells. Here, however, you can see that the algorithm, you have still, the cells are clearly a number that's a bit reduced, but you have still a really single cell behavior and cell separation, which is what you want. Now, if you get to the bad point, you can see here that now the cell calling algorithm has really great difficulties, and you can kind of understand why as well, because looking at the graph, the way it looks like, you could easily either this could be your we'll put it between quotation marks, but your steep cliff followed by the plateauing knee, but it could also have been this, and so this creates really big problems because it also results in loss of single cell behavior. You can see that the cells are not nicely separated, and there's a really big clump of them. And so I guess our next question is then, all right, if I get to this point and I also want to continue and looking, I still don't want to abandon ship and I'm doing some sort of analysis, then I can ask myself, okay, well, what does the bad look like? And can we turn it into something good? Generally, if we look at what that looks in, for example, a UMAP plot, we can see that the problematic cells tend to obviously clump together, and this can be a result of either ambient RNA, dying cells, or a whole other plethora of issues. And now, for example, if your data is, was made of really high quality cells, like for example our mouse data, what you do is you go through a few filtering steps, you can visualize it, you can already see that there is very nice separation within the cells. You could continue in seeing, okay, well, are there any doublets? And you can see that in this data there are very little number of doublet cells. And then you can actually really, just with a few filtering steps and optimization, you can really proceed to analysis. However, for our human data, this was very different. So we started looking a little bit into filtering. We can see that the two different groups here are the treated and the untreated groups. They're very dissimilar. And after we did the separation, as you saw before, we get this clump of cells that is really, really problematic. And now again, usually what you can do is go through different phases of filtering with different types of filtering and different types of optimization, and you just visualize it. Here you can see that also going into the how many doublet and single cells, we can see that there was a lot of doublet cells as well, and this was very problematic. Now sometimes what you can choose to do is, rather than going into this infinite loop of filtering and trying to optimize as much as possible, you can also choose to temporarily proceed with analysis. And the reason for this is that, because analysis can help you understand something more about the biology, what is actually happening to your cells. And it can also be sometimes indicative, give you some sort of indication of which direction to take to further filter and optimize your cells. And so this is kind of something that we did. I went and looked into some of the fundamental markers that were very central to the type of biology that we were studying. And we can see that some of the markers that needed to be there were there. And then we saw something else kind of interesting as well, is that one of the subclones that wasn't really identified was expressing a really high amount of TGF-beta1, which is really important to the type of study that we were doing. And so this is kind of exciting because it also helped us to get a better idea of what was happening to those cells and whether there were any of those subclones that we can continue using. And the exciting part of single cell data in this regard is that you can always look at specific clusters, and these clusters can, you can hone in on the cluster. For example, here you can say, okay, I just want to see the DC cells and the CD14 monocytes, and you can go and say, okay, well, how about I look at this specific cluster, I go into further separation, and then continue asking questions, for example, which genes are specifically differentiating this cluster, etc., etc. And you can also, for this, go into an iterative process. Obviously, if you have very low quality data, you have to do this very carefully, and there's many considerations to take, and it's not something that you could go straight to for a publication. But in trying to understand what the biology is that you're looking into and how to further filter the cells, because this could be very useful. Sometimes you also have something else that I really love in terms of some of the R packages, and those these type of plots here. And so this allows you to look at subclusters that are informing your clusters. And the reason why I find this super exciting is that you can ask yourself, right, well, for example, which subclusters are feeding into the bigger clusters, and you can look at this in the context, for example, of clonotype tracking. If you're doing, for example, multi-omics data integration or cellular trajectory analysis. And for those of you out there that work, for example, either with cancer cells, but especially with immune cells as well, for instance, T cells there can be many different types of subtypes of T cells, and each one of them have a very distinct function. And so looking into subclusters can be really informative and can give you some very exciting data in that regard. So that being said, I think this kind of takes us towards the end of this single cell data adventure, and I'm happy to take any questions.
Thank you, Jennifer. I'm really always excited to see what you're coming up with all these single cell experiments. I think we had a hand raised, but not anymore. So for everyone, please feel free to put questions in a chat or raise your hand if you would like to ask the question yourself. Okay, we have a first question from Ruccio Fino. What kind of resolution can be expected to identify gene variants at the single cell level?
Resolution of in terms of how deep you can go into messenger RNA?
Yes.
Well, that's a really good question. The resolution is dependent on whether you have managed to obtain the proper amount of sequencing depth. Now, and this is I find a really, really good question, because it was one of the things that I was asking myself as well. It depends on. I find that often when I ask a question surrounding single cell, I often get the response of "it depends." And in the beginning, that used to frustrate me a lot. But it kind of, like, as you go further, you realize that "it depends" it makes sense in a way that is specific to the biological context that you're studying. For example, if you go, we work a lot with PBMC. So white blood cells are usually, I think, and Jerry can correct me here if I'm out with the percentages, but you're really looking at to a 60% to 80% sequencing saturation with these types of cells. So in terms of resolution, if you are looking into genes that are super rare, then you're going to have to sequence deeper. And if you're looking for genes that are very highly expressed, then your sequencing depth, can be a little less. And now this, on one side, it sounds very vague, but I actually, if we have a little bit of time, Martha, I have a slide that might elucidate that a little bit better. And this is, and there you go. This is one here. Okay, so to answer the question, right? If we look here at our expressed genes, the highly expressed genes generally have the highest chance of being caught by your Illumina sequencing platform, right? And so if I have a highly expressed genes, chances are, it will mostly be captured by the platform. If I, however, have very lowly expressed genes, the chances of this being bound to the chip and being sequenced is much lower. And so what that means is that if you want to have a sequencing depth, a resolution that is higher for genes that are lowly expressed, you're going to have to increase your sequencing depth. If that, on the opposite side, if you're dealing with genes that are not that highly expressed, then your sequencing depth does not have to be that deep. So to answer your question, the resolution can be extremely high. It just matters how deeply you're sequenced and whether you know that you're dealing with genes that are rarely expressed, because that's the other problem as well. If you don't know that, then you usually end up with a standard sequencing depth that any sort of genomics platform uses, and you might miss out on them.
I might quickly jump in here because I'm not sure potentially if Nochio is asking about gene isoforms. Are you? Yes.
Yes.
So because the regular 10x workflow deals with short read sequencing, you are not necessarily going to detect gene isoforms. However, our assays, 3' and 5' assays, are compatible with long read sequencing as well. There are some modifications in the workflow, but it is compatible with long read sequencing as well. So if you are interested in looking at isoforms, that would be the way that you would go.
Yeah, we are always happy to also organize a chat to dive deeply into any project ideas at the very early stage. So that was our question. Feel free still, our dear attendees, to put some questions. I just wanted to clarify something that Jennifer said. So if your sample is really bad and you would still put it and want to run it on the machine, you will not destroy the machine. You will clog a chip on the instrument, but the machine will be fine. So you don't have
really? It doesn't. I was like, I was almost scared. I was like, right, this is a microfluidic system.
It's good to be scared, but it's not that bad.
Everything stays on the chip, basically.
Okay. All right. Well, that's good to know. Thank you.
You're welcome. That's why we're talking about cells here. We have another question. What is the lowest number of cells that's worth running in the Chromium platform? I think that will be more for myself and Jerry. So basically, minimum that is advised or I've seen is like 500 cells, if I'm correct. The thing is that when you load cells onto the instrument, the efficiency capture is around 80% with the newest iteration of the kits. So you will, if you put 1,000 cells, you will have 800 cells back. And because of the cost of the assay, it is always recommended really to use it to its fullest, and perhaps use methods how to maybe put few samples together. But that's also, Jerry, want to add something to that?
Yeah, I mean, you can essentially go as low as you want, but it just becomes a point of diminishing returns, right? So it really depends on the ultimately your experimental question. We usually recommend 500 to be the lowest, but we have had people go lower. It's just that, if you're sequencing 200 cells, is that really worth it for a single cell data?
Yeah.
And also, I guess the other thing to add here is that the more cells that you target for capture, the more you'll have to sequence. The less cells you target for capture, the less you'll have to sequence. So it is also about striking that balance as to what you need versus, what you want.
Also, the technology, it actually allows you to distinguish between heterogeneous cell populations. So if the lower number of cells, the heterogeneity might be lower because you're capturing just a part of your cells. So if you have 20,000 cells, although they could be same types of cells, that wouldn't make any sense. But yeah. So
yes, and yeah. And then also regarding to your follow-up question, that we do mean 500 good cells. If you have lower viability or debris in there, which makes your counting inaccurate, that will ultimately affect your final capture efficiency and how many cells you ultimately recover.
Yes, because if you have 500 cells and then there will be debris, you will end up with some Jennifer bad examples where you will have this blob that you will not be able to distinguish this 500 cells that you need. So that's why we always highlight that the cell preparation is the most important part of the workflow. Jennifer, do you have any advice for new users of 10x Genomics, new budding bioinformaticians? Because I think you were never really trained as a bioinformatician. Is that correct?
No. So I think for anybody who wants to embark in the single cell adventure, I would strongly suggest to apply for the single cell fellowship you guys are offering, because that was really helpful to me in the beginning. To have, I find it super useful that, and this is, the support that 10x gives. So you have a question, either Jerry or Jeff, to just discuss the technical details. For us, I had a lot of conversations in the beginning with Jeff as well that, especially because we didn't know, like, what do you do? Do you, freeze it down? Don't freeze it down? Do you have mice on one side? You have this on the other side. And so you just you have to, think about your workflow. I think that is to answer your question succinctly. Think about your workflow, because the workflow and the stress that you undergo in the lab affects what the quality of your cells are going to be. Could affect what the quality of your cells are going to be. Meaning that if the protocol is not clear, or errors are made, or there are situations in which you have to things aren't done in a way that is, for example, conform to the 10x protocol, then the more your cells are sitting around waiting, the worse the quality is going to be. And this I can specifically say to, for our own cells specifically, whether they are live cells or that haven't been frozen down, or cells that need to be defrosted. Cells can be very finicky. And so I would really, really strongly think about the workflow, and even just it's hard to say dedicate some time to just do a pilot experiment, with one sample with 10x reagents. But what you can really do, for example, we didn't have that option, but the option that we did have is just do the workflow up until the point that you have to load it. You have to count the cells to start loading on the Chromium controller, because that is a lot of cell counting and recounting and adjusting. And that makes a really big difference for what it's going to look down to. So if you have a very streamlined process in which your cell count and validation and putting everything together up until the point that you start using the reagents, you are a big step ahead already. Bioinformatically, there is the only thing that I can say is have a chat with fellow people who do bioinformatics and single cell, because there is a million different ways of doing this. With this cell blob, I've talked to people that went like, no, throw all the data out, and people who went like, oh no, you can try to do it this way. And you could all like, if you look on the internet, there's always very different ideas about this. And so every idea is different because it is, again, dependent on what the biological context is of your research. So, but for me, it really helped to just talk to people and figure out different ways to analyze and visualize, really.
Thank you, Jennifer. That was really fantastic suggestions from you and fantastic presentation. We are now on time, so we will be wrapping up. Thank you, everyone, for attending session today. Thank you, Jennifer, for presenting. Always a pleasure to talk about your research. Thank you, Jerry. And stay tuned. This series will be coming back in a few weeks, so check your inboxes